All right, it's Riffy. I'm here to start up the Unit 3 notes, Skeletal System. Part 1B notes, we left off talking about the different types of bone tissue, specifically compact and spongy bone. Now we're going to talk a little bit about the cells, and we're going to get right into how your bones actually grow starting just shortly after fertilization. So if we were to get into the bone cell types, things you're going to come across, particularly when we start talking about bone growth and development, there's going to be three kinds. You're going to have what's called osteocytes. Osteocytes are the ones that are going to refer to the most because they're the most common that you're going to find in bone tissue, but they are the ones that are mature, non-dividing osteoblast cells that will forever remain trapped within the lacunae of bone tissue. Osteocytes function to constantly maintain and repair your bone, and they do what is called calcium recycling. So they remove calcium from the blood, they put it back in, they remove it from the bone, they put it back in the blood, back and forth, back and forth, back and forth. But most of the bone cells that you're going to have in a human body are going to be osteocytes. Then we have two other kinds, and what they are mostly involved in is what is called bone remodeling, and we'll get into that in just a little bit. But osteoblasts, I always remember osteoblasts because of the fact that it says osteoblasts and it begins with the B. They are the bone cells that build bone. Osteoblasts build. They are bone cells that produce what is called osteoid, that essentially makes the dense, hard, rock-like substance of the bone matrix. When during bone formation, when osteoblasts become so you could look at it like so involved with their job that they actually become trapped. They trap themselves in that calcified bone matrix all around them. When they become trapped, they're there forever. When they get there forever, they end up changing or differentiating into osteocytes themselves. The second kind are called osteoclasts. Osteoclasts are the ones that actually do the dissolving of bone. They are extremely large, multinucleated bone cells, and what they do is they actually digest or kind of scrape away or eat away or erode all the bone cells and gives bone their shape. The calcium matrix that eroded away increases the level of calcium and phosphates in the blood, so they dissolve and they put calcium and phosphate into the blood, and that actually can be traveled and used in other places. So remember how we said that one of the main functions of the skeletal system was to store minerals? This is where it's stored. So these osteoclasts are what release them and allow them to travel throughout the bloodstream and be used in other places. So again, keep in mind, osteoblasts build bone, osteoclasts dissolve it, and osteocytes are the most common bone cell types. So that leads us into our first entrance card question for 1B. And what we're going to get into is kind of the function of osteoblasts and osteoclasts as you get on with life. So if you solely thought about the function of osteoblasts and osteoclasts, why do you suppose there is a large percentage of senior citizens that it develops osteoporosis? Osteoporosis is essentially a bone degeneration disorder that weakens your bones. So if you thought about the functions of osteoblasts and osteoclasts, why do you suppose a large percentage of senior citizens develop osteoporosis later in life? And it definitely has to do with those two types of bone cells. Just to review really quickly before we start getting into bone formation, I want to review the basic types of tissue that are in bone. If you look out here, this is that wedge that you will find from any kind of flat bone or long bone or anything like that. But remember, on the outer edge, you're going to find the periosteum. And then on that inside portion here, you're going to have that compact or dense bone, keeping in mind that osteons are the repeating formation for that. And then once you get a little bit deeper inside that bone, you will notice that it turns into spongy bone, and the repeating units in spongy bone are called trabeculae, keeping in mind again that spongy bone has a lot of space within it. Compact bone does not. Periosteum on the outside. Periosteum is also where the blood vessels start to embed themselves into the bone tissue and start forming, perforating in central canals. Alright, so bone growth and development is not an easy topic. It's actually going to be divided into two categories, and it is so detailed that I'm going to try to give you the meat and potatoes version, or the version, I would call it the high school version. But I can't do that without giving detail. So I'm going to tell you what you need to know, and I'm going to also give you extra detail, but only concentrate on when I really specifically say, this is what I want you to concentrate on, or what I want you to know. So bone growth and development as a whole, basically when upon fertilization, 6 weeks later is when the skeletal system actually starts to form and bone growth occurs. But it starts off as cartilage as a whole. So if you were to look at it, a human being after 6 weeks after fertilization, it would look like this. So it has the shape of a human being or an alien, however you want to look at it, but it has the shape of a human being or an organism. But what you can't see are in fact bones anywhere within this thing, with the exception of a little bit of ribs here, a little bit of sternum forming. You can see the shape of the skull, but there's no definitive bones. So how does this start to occur? Well, it happens through a process called osteogenesis, and then osteogenesis is just the creation of bone. And during that development, in both types of bone formation, cartilage is replaced by the bone matrix in a process called ossification, and there's going to be two kinds. There's going to be what's called intramembanous ossification and endochondral ossification. So trying to think of the best way to relay this to you, what I did is I stole some videos from YouTube. What I'm going to do is I'm going to play them, let the sound play out, and I'm going to pause them and point out specific things that I think you should pay attention to for intramembranous ossification. This is going to be the first one we're going to cover. One thing you definitely need to make sure you understand, intramembranous ossification is typically going to occur in your flat bones. These are things like your scapula, the pelvic girdle, or the coccyx. You have the mandible, which is your jaw, and the clavicle. So you have the flat bones of the skull, mandible, clavicle, scapula, which are your shoulder blades, and the really broad hip bones called the coccyx.
Intramembranous ossification results in the formation of bones, specifically the flat bones of the skull, the mandible, and the clavicle. To begin, intramembranous ossification takes place in mesenchymal or fibrous connective tissue. In the connective tissue, mesenchymal stem cells come together or aggregate and then begin replicating.
Okay, you don't necessarily have to worry about memorizing the name of mesenchymal stem cells. What I need you to know is that when the bones of your cranium are starting to form, you have very specific cells called stem cells. Stem cells essentially are cells of the human body that have not been told what they're supposed to be yet. So when these mesenchymal stem cells come in, they haven't been told that they're going to be bone cells yet until a process that involves differentiation starts to occur, where the genes start getting turned off in those cells. And as they get turned off, the ones that are left on are the ones that say, hey, you're going to become bone cells. So when that happens, those stem cells differentiate into bone cells themselves. And this is all occurring in connective tissue.
After sufficient replication, these mesenchymal cells begin differentiating, becoming bone-creating osteoblasts.
Remember, we said osteoblasts are one of the three types of bone cells, and their main job, osteoblasts, build bone.
These newly formed osteoblasts then release an uncalcified bone matrix known as osteoid.
So when they talk about uncalcified, basically you've heard the whole saying, milk does a body good because milk has calcium, and you need calcium to build strong bones. Well, it starts off at, at that 6 weeks of birth. So if this were your skull, those cells form this nice ring with one in the center here, but it's uncalcified. So it's osteoid, which is like the beginning of bone tissue, but it doesn't have that hard, dense calcium structure to it yet.
The osteoid is then calcified with the addition of calcium salts and other minerals. The enzyme alkaline phosphatase acts as a catalyst in this process.
Okay, you do not need to know anything about alkaline phosphatase, but what you need to understand is it has phosphate and it combines with calcium, and what that does is it starts to form the hard matrix of bone. If you take calcium and phosphate, you have that calcium phosphate mineral that I had you read about in our lab notes. And what it does then is the cell uses both of these things and it starts producing that hard, dense bone matrix around it. What is going to end up happening if it continually produces all this stuff around it? It's like building a jail around itself, and that cell will get trapped in this little area. Well, that little area, if it traps inside of itself, that area becomes the lacunae, and this osteoblast, since it is now trapped, becomes an osteocyte.
As the animation portrays, osteoblasts, when surrounded by calcified bone matrix, can differentiate further into osteocytes, which house themselves in hollowed-out living spaces called lacunae. These cells support the bone in maturity. This first process is known generally as ossification, and where ossification takes place is known as an ossification center.
So I want you to imagine that this is one ossification center that is starting to produce the skull of a 6-week-old embryo. So if you're looking at this, you want to imagine that this ossification center is actually occurring in several centers throughout the entire region that is going to become the skull. And it forms in very specific spots according to the different bones of the skull. So this little center happens in several places throughout that one bone. Let's say it's the frontal bone of the skull. And what will happen is it continually produces this bone matrix until ossification centers actually start to touch each other.
As intramembranous ossification continues, osseous branches, known as spicules, are formed. These spicules will eventually touch one another and fuse together.
Now what ends up happening is this is the beginning formation of what are called trabeculae. And trabeculae are the groundwork for spongy bone. So when your bone develops, particularly these flat bones, it starts off as stem cells that become osteoblasts that start producing the bone matrix. Those osteoblasts become trapped, they become osteocytes. They continue to produce the bone, and it produces in an outward range of motion until they start to touch, where they become trabeculae. So basically, the beginning formation of bone actually starts off as spongy bone. What this is supposed to represent here is a blood vessel. What you're going to notice is as these spicules form more and more trabeculae, those blood vessels become trapped within the bone matrix,
forming a larger piece. Seeing as mature bone is highly vascular, spicules will grow themselves around blood vessels. This also allows for increased nutrient and waste exchange during these crucial developmental stages. The initial bone formed from intramembranous ossification is known as spongy bone.
So I know this guy in his video, he kind of jumps from, hey, we just formed one trabeculae to, boom, a whole bunch of spongy bone. So what you need to understand is there was a whole bunch of ossification centers spread throughout this entire region, and what actually has happened. So maybe I can draw some. So maybe there was an ossification center here, and an ossification center here, maybe an ossification center here, and one right here. What has happened is as they spread in their bone growth throughout, they start touching more and more and more and more and more and more different spicules until you get this entire spongy bone matrix throughout the entire. Now, if you notice, we have no compact bone yet. So you have this layer on the outside, you have this layer on this outside, but all we have in between is spongy bone.
Due to its sponge-like appearance. Through the process of bone remodeling, the osteocytes at the edge of spongy bone can reorganize themselves into tightly packed bundles known as osteons.
Okay, we are going to get into bone remodeling a little bit, but basically what happens is, since this spongy bone is now formed, you have more osteoblasts, and now you have osteoclasts that come in. The osteoclasts are going to dissolve some of this trabeculae or the spongy bone, and then the osteoblasts are going to use what they dissolve to reform it into tighter, densely packed layers of compact bone here and compact bone right here. And then you know that the repeating units of compact bone are actually called osteons. Osteons are only present in compact bone. Trabeculae are the repeating units in spongy bone.
These osteons are the foundational building blocks of compact bone, which is then formed on the outside edges of the spongy bone. This layering of compact bone, spongy bone, and compact bone again is the defining characteristics of bones categorized as flat bones.
You can also look at this as the characteristic of the diaphysis of long bones.
Through a different process of bone remodeling, the middle layer of spongy bone can be removed, creating a medullary cavity.
This is where long bones come into play.
The final structure of completed bones is the outermost layer of connective tissue, known as the periosteum. The innermost portion of the periosteum is composed of fibrous connective tissue. In this tissue, more osteoblasts can be produced, allowing for the bone to grow in width. This is known as appositional growth.
So basically what he was saying is Once this compact bone is formed and this compact bone is formed, and this blue represents the periosteum on the outside of each side of that flat bone, underneath there are still osteoblasts, and those osteoblasts will continue to produce bone matrix in an outward direction in both sides until it becomes thicker and thicker and thicker. So what I want you to see now.
Case of flat bones, like those of the ribs, scapula, pelvis, and skull, the ossification process is known as intramembranous ossification.
Isn't that a cute baby?
The process is initiated by
cells that have not been told what they want.
mesenchymal stem cells, which are cells that have yet to be differentiated into a particular cell type. Eventually, centrally located mesenchymal cells within the embryonic fibrous connective tissue differentiate into bone-forming cells or osteoblasts. This process constitutes the formation of what are called ossification centers.
So this would be the uncalcified.
In the matrix formation phase that follows, the osteoblasts begin to lay down osteoid, which is the organic part of the bone, consisting of collagen fibers. Some of the osteoblasts are entrapped within the osteoid and are then said to be osteocytes. The osteoid then calcifies and forms slender, needle-like structures of spongy bone called spicules.
Spicules!
Which aggregate in the form of small supporting
Boom! So you go
beams
from
or
spicules, they start
trabeculae.
touching each other into trabeculae,
Meanwhile,
but
the
they're really
blood vessels
thin.
on the outside of the spongy bone condense and form the periosteum, the fibrous sheath that. In the case of flat bones, like those of the ribs, scapula, pelvis, and skull, the ossification process is known as intramembranous ossification.
Now,
The
this
process
is a step that we
is
didn't have in the
initiated
last
by
video. So
mesenchymal
this would be
stem
the step
cells,
between the formation of compact bone and after the formation of spongy bone.
which are cells that have yet to be differentiated into a particular cell type. Eventually, centrally located mesenchymal cells within the embryonic fibrous connective tissue differentiate into bone-forming cells or osteoblasts. This process constitutes the formation of what are called ossification
Come on, you know what
centers.
this thing's called?
In the matrix formation
right, it's
phase
called
that
an
follows, the osteoblasts begin to lay down osteoid, which is the organic
Boom!
part of the bone, consisting of collagen fibers.
And that is intramembranous ossification. So let's review. Intramembranous ossification, the bone develops within sheets of membranes of connective tissue that kind of resemble cartilage. The bones usually formed in this process are those thin, flat bones, skull, jaw, clavicles, costia.
Some of the osteoblasts are entrapped within the osteoid and are then said to be osteocytes.
The process starts at
The
an
osteoid then
center,
calcifies and
is
forms
completed by bone cells
slender,
called osteoblasts,
needle-like
which are very
structures of
condensed
spongy bone
summary.
called
And those
spicules,
osteoblasts
which
produce a new bone
aggregate
matrix
in the
while
form
at the
of
same
small
time
supporting
promote
beams
the
or
formation
trabeculae.
of that dense calcified bone. The osteoblasts continue to remove calcium and react it with that
Meanwhile,
enzyme,
the blood vessels
and
on the
continue to
outside of
form
the
that
spongy
nice,
bone
dense bone
condense
around it. But
and
then what
form
happens
the
is
periosteum,
the osteoblasts become
the
trapped
fibrous
or trap
sheath that
themselves
covers
in
bone.
lacunae, and they turn into
As the trabeculae thicken, an interconnected network forms, resulting in what's called woven bone.
Those spicules start to touch. Boom! You get what are called trabeculae, which is the framework for spongy bone. The trabeculae start to get thicker and
Which
thicker
is
and
characterized
thicker.
by
Once
a
that
somewhat
occurs, the
haphazard
outer
organization
edges
of collagen
in the
fibers
remodeling
and
process,
is
turning that
mechanically weak.
trabeculae and that spongy bone into compact
Eventually,
bone,
this
and then
leads
boom,
to the
you have a bone
formation of
sandwich. You have
lamellar
compact
bone
spongy
around
bone,
the newly
compact
formed
bone on the
spongy
bottom,
bone.
and you start
This
to
lamellar
develop
or
skull
compact
bones.
bone has regular parallel alignment of collagen in the form of sheets.
Continues and continues and repeats itself. Here is a very interesting
Or
picture of a fetus
lamellae,
of 12 weeks
and is
where
mechanically
you can
quite
actually
strong.
see the skull bones themselves forming the trabeculae of spongy bone. So these skull bones here haven't even reached that step of forming compact bone on the top and bottom edges. It's a crazy picture. What's even nice about this is it has the flat bones of the skull, your jawbone here, which starts to be a little bit more defined. You can actually see some of the compact bone. But this picture to me is mind-blowing. And this picture to me is quite boring. Alright, so this brings us to our second entrance card, and actually it's the only other entrance card for Part 1B notes. So what you have here is, I'm going to give you the steps of intramembranous ossification, but they're going to be missing some words. At the top of the next slide is going to be your word bank. You want to take the words from that word bank and fill in the missing words in the steps of intramembranous ossification. Alright, that's going to be the end of Part 1B notes, intramembranous ossification. As always, please, please, please utilize me as a resource. Ask me as many questions as you want. I appreciate everything you do. Thanks.